Hello, everyone. My name is Gregory Reeves. I'm a postdoc at the University of Cambridge, and I'm very honored to be giving this talk to the JR Biotech Foundation about an introduction to molecular biology. You can contact me by the email listed below in case you have any questions about this talk. I'm very happy to receive anything. So today's topics are going to be including a few things. We're going to be discussing the polymerase chain reaction, or PCR, gel electrophoresis, DNA or RNA sequencing, genotyping organisms, particularly plants, and then gene expression analysis by quantitative PCR, and then we will briefly discuss gene editing with CRISPR-Cas9. So I just wanted to bring everyone to the same starting point about what is DNA. DNA is a biological macromolecule that forms a double helical structure that everyone, I'm sure, is well aware of. This helix is formed of a sugar phosphate backbone with 4 different nucleotide base pairs. And these base pairs are given the names adenine, thymine, guanine, and cytosine, or ATGC. As seen, just a moment, pointer options, laser pointer, as seen here on the far left. The base pairs in DNA form hydrogen bonds with each other. So adenine and thymine, A and T, form 3 base pair, 3 hydrogen bonds between these 2 bases. And then guanine and cytosine form 3. So 2 and 3 hydrogen bonds between the 2. And these are bonds between oxygen and hydrogen, or nitrogen and hydrogen. The consequence of covalent bonds forming the backbone of DNA versus hydrogen bonds forming the bond between the 2 helical strands means that it is heat sensitive. And that is that the hydrogen bonds can be broken down by heat, but not the covalent bonds. So strands can be separated with high heat, but not the backbone itself. That requires a chemical process. Before going further into that detail, the base pairs in DNA encode amino acids, and amino acids form the structure of proteins, which perform many biological processes. In fact, nearly all. In this diagram, you can see that from the genetic code present in DNA that gets transcribed into RNA, that gets translated to protein, we arrive at this sequence. For example, a gene sequence with GAA will encode glutamic acid, or a sequence encoding CCC will be proline, the amino acid. And in nearly all organisms, this code is universal. There are a few microorganisms which this is not the case, but for higher organisms like plants or animals, this code is nearly universal, which has a huge utility in applications. The biggest application for this is the PCR reaction, or polymerase chain reaction. It's probably the most useful tool in molecular biology, and it forms the basis for most techniques in molecular biology. What happens is that in your target organism, like a plant, crop species, an animal, you would like to understand the sequence or molecular markers at a given position. But you have to derive that information from the entire genome. So here in this diagram, we have in this sort of yellow-orange color a region of DNA they were interested in amongst the entire genome, which is colored in gray. And it could be very much more DNA than is pictured here. That DNA is heated up, that liquefies those hydrogen bonds, and then we add these synthetic or man-made or ordered primers. So those are our short single-stranded DNA molecules that are complementary, or they match the sequence we want to that region we're interested in. And we include two of those. One to attach to or anneal to the 3' strand of the DNA, or one of the helical strands. And then we have another one that's complementary to the flanking area of the other strand of DNA. And the reason we have two of those is because, well, will become obvious later. But we add then thermostable polymerases and nucleotides to do DNA replication in test tube. From one primer, the DNA can be extended in one direction continuously, and then from the other primer, the DNA can be replicated again in the other direction. If this process is repeated again and again and again, we can just continually replicate that area of interest, so this orange-yellow area. So if we repeat these steps, then we end up creating numerous copies of that area. And after, like, say 25 repetitions of this, we end up with not only one copy of that sequence, but 1,000,000 copies. So in normal biological applications, we'll do 30, 40 cycles, and that will create billions of copies, in fact. PCR is becoming more affordable, and but the fundamental requirements are we need template DNA, so that is up to you to create. So from your organism of interest, animal, plant, or even fungus, whatever, bacteria. And I've just put some links here for like low-cost DNA extraction protocols for plants or fungi. If you're interested in, like, for example, like a pathogen. DNA polymerase, this is the enzyme that copies the DNA, is quite important. And again, I've included a link for a really cheap one that has high fidelity, that is even useful for sequencing applications, which we're going to get into later. You usually need divalent cations, that is metals that have a positive charge, and this is usually manganese. Um, what's really important is DNA primers. These are short DNA segments that target the area of the genome of the organism you're interested in order to replicate that area. And this aspect of PCR is probably the most critical because it requires your expertise, your knowledge to design. And so I have included two tools here, Primer Design and Primer 3 software, which are both free and have YouTube tutorials on how to use, so that you can design primers for DNA segments that you're interested in. You also need DNTPs, so these are the nucleotides that are going to be used as the building blocks for the DNA template. And then you need water as a base. Additionally, PCR can be very complicated, and you can have additives that alter the binding properties of your primers, or mostly that, like DMSO, formaldehyde, glycerol, etc. So it depends, and the reason why you would use those is if your organism has many hydrogen bonds, so a high GC content. PCR can be inhibited by a number of compounds, and there's a list here. Not going to get into them. But a lot of them have to do with how clean your DNA is to begin with. So a lot of these compounds that are involved in DNA extraction can inhibit subsequent steps involved in analyzing that DNA. So keep that in mind. You can optimize PCRs that go wrong through a number of ways. PCR sensitivity can be improved by, for example, increasing the concentration of primers, increasing the concentration of the polymerase, increasing or decreasing your temperatures at which you do the thermocycling process. You can let me just move my. You can increase the specificity through these steps, like reducing the temperature, the time it takes for these temperature steps, or decreasing the number of cycles, or using what's called a hot start technique. So that's using a polymerase that has a that is only active once it's been heated, or modifying your concentrations. It all actually becomes quite complicated. But once you have a successful PCR, you would want to analyze that sequence that you've created. One of the most useful tools is a technique called gel electrophoresis. And it's used to separate mixed pools of differing RNA and DNA based on their electrical charge. And this electrical charge is more or less proportional to its mass, which infers its length. Because DNA is a linear polymer. And if RNA is your nucleotide or nucleic acid of interest, it can be based on shape. If it's in its native form, or if you denature it or remove that, what they call secondary structure, it can be separated based on mass, and that depends on the chemicals you include in your gel electrophoresis. So let's just go into gel electrophoresis. Here is sort of a diagram from the side of what they call a gel electrophoresis tank. Here in this gray box is a gel matrix. This is usually made out of agarose, which is a it's like a gelatin or a thick substance made out of agarose, which comes from seaweed. You melt it in a microwave or at high heat, and then pour it into a casting mold. And in that casting mold, you include like a comb or a device to have wells. And then into those wells, you deposit DNA within a buffering solution. So that's water with various salts dissolved. And because DNA has different lengths that you might deposit, and because DNA has a negative electrical charge, once you apply electrical current within the system, DNA will begin to move into the gel matrix. And the gel matrix is designed to separate DNA based on its electrical charge. So small DNA segments like this one here will migrate very quickly through this gel matrix because it has less drag. A medium-sized fragments will travel slower, but more quickly than large fragments that have a lot more friction with the gel matrix. And again, this involves applying electrical current. So the side where you apply the DNA has a negative charge of electricity running through it, and then the migratory side has a positive charge. If RNA is added to this sort of matrix, you can infer how much structure it has, like how much folding, based on this migration patterning. If you add denaturing agents, so agents that break down hydrogen bonds, you can then infer the size. In general, this is based on size, and that infers how large your DNA fragment is. So here is a real-life example. This is a gel electrophoresis that I've created. And again, in these gels, you include some sort of compound that can bind to DNA, that can fluoresce under some sort of a light form. So the most common way of doing this is including ethidium bromide, which is fluorescent under UV light, but only when bound to DNA. Another common one is cyber green, which fluoresces under blue illumination, but also UV works. And that is a way of identifying areas in that gel matrix where DNA is present. So if you can sort of see in this picture these slots, these marks here, And those are the areas of the gel where the PCR product was inserted. And then voltage or electrical current was applied to this gel, and that DNA migrated from these wells, or these areas where DNA was inserted, downwards towards the positive current. And then we then viewed this under UV illumination after some time, like after a couple hours, like 2-3 hours. I've circled some areas here in red, and those areas are indicating basically PCR products with the same primers, but they're amplifying the same genomic region, but in different plants. And these plants actually are exhibiting polymorphism. So they have a different DNA sequence at those regions. So for example, here at this particular region, these plants here produce 2 bands with that same PCR primer pair, whereas this plant produces just 1 band. And again here, we can see like a very intense band with that primer pair there. And in a different plant, we can see 2 different primers. You can probably identify other differences, or what we call these differences are polymorphisms. And so these polymorphisms are very useful because if we use statistical analysis to correlate these differences in DNA sequence to our trait of interest, then we can learn more about where those genes that control that trait are. So this is a real-life example of a disease resistance locus or position in chili peppers. So I ran these gels. These are disease resistance genes in chili peppers that control resistance for a fungal pathogen called Phytophthora. And we can see that all these lanes are from different individuals. A lot of the individuals are very similar to each other, but some are different. And the ones that are different actually had resistance to the pathogen. So these tools, once you identify for example, a primer pair that correlates to your trait of interest, we call it a molecular marker, which we're going to get into a bit later. You can also identify molecular markers by sequencing, which is what I'm going to describe right now. DNA sequencing is really a lot more precise than gel electrophoresis because you can identify even more differences in the DNA sequences themselves based on, instead of basing your differences on size, which can, which are more like large-scale differences, actually. You know, this might be a difference of 50 base pairs, so 50 DNA sequences or base pairs are differing between these 2 samples. But what we know now in biology is that a lot of biological differences actually depend on far less difference. Even a single DNA base pair can influence a trait dramatically. You can sequence RNA as well to look at gene expression differences. But ultimately, that needs to be converted to DNA before it can be sequenced. So this information is useful for both applications. And again, numerous methods for sequencing exist. Some are cheaper than others. It depends on a technology called chain termination, which is the original way of sequencing DNA developed in the 1970s. We know that DNA has a sugar phosphate backbone. So this gray P is a phosphate group. This purple, I would say hexagon, but it's more like a pentagon sort of thing, is the sugar. And that's a repeating structure in both DNA and RNA. But on that sugar structure, which is either ribose in RNA or deoxyribose in DNA, has a 3 prime. So for example, on the third sugar, on the third carbon, there is an oxygen and hydrogen, or they call that a hydroxyl group. You can add a new base pair on. And a new base pair consists of a base pair, which can be adenine, thymine, guanine, or cytosine. That sugar, another hydroxyl group attached to the third prime sugar or carbon, and then 3 phosphate groups. But you absolutely need that 3 prime hydroxyl group. So you need that oxygen there. If oxygen is lacking at that position, you cannot add a new DNTP, they call it, or a new base. So they call this chain termination. Means you can keep adding onto this chain of bases, like a chain of sequences, but the moment a base lacks a hydroxyl group, nothing else can be added there. So it will just stop. In order to accomplish this in real life, you need to run a reaction with the following components. You need a DNA template, which is what you want to sequence. You need primers, which are basically the same primers you use in your PCR reaction, but you'd only use one of them because you're only sequencing one strand of your DNA. You need DNA polymerase. Is almost the same as PCR, and you need this is a more modern approach, but you can also use your DNTPs or your nucleotide building blocks can be labeled in some way or another. That can be like radioactive or fluorescent, or they can just be standard nucleotides, but you need to run them on a very clear gel. I'll show you this. But the fifth component, so all of these are more or less very similar to a typical PCR reaction. The fifth requirement is modified nucleotides that don't have that oxygen, that hydroxyl group. So you need a small proportion of nucleotides that have this just hydrogen at the end. And those modified nucleotides will terminate the DNA strand from elongating. So the way that looks, like for example, if you just put normal nucleotides and a small proportion of modified nucleotides, instead of and ran those on a gel electrophoresis that was capable of differentiating lengths of DNA that differ just by one base pair, you would see something like this. So for example, here is a very simple gel wherein we ran 4 different PCR reactions. In one reaction, we included all the nucleotides for PCR, but we added what we call DD ATP. So this is adenine, so this base pair that has a hydrogen at its 3' end, as opposed to normal DNA, which has a hydroxyl group there, or an OH. So that will cause a chain termination. So any replicating DNA strands that incorporate that base pair as its final base pair will just stop. So it will create a band at that position. For example, here then next, in a different reaction tube, instead of adding the DD ATP, we added DD GTP, which is the guanine equivalent. And here we can see that using the same DNA template in all these reactions, but with 4 different tubes, each tube containing A, G, C, T, with these modified base pairs, we can see that the reaction has terminated or stopped at very discrete positions. So if we work our way downwards, we can see that at the 12th position, there's a band in the A lane. So that means the sequence is A there. Then 11, it's no A, no C. There is a G, there's no T. So that means there at that position is a G, or guanine nucleotide. In this 10th position, we can see there's no A, there's no C, there's no G, but we do see a band appearing in the thymine lane. So that must mean that that 10th position is thymine. And again, we can work our way down, 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 and then we can reconstruct the DNA sequence. So we have A, G, T, C, G, A, G, C, and so on. So using these 4 separate reactions, we can work out the DNA sequence. This requires us, though, to use what we call polyacrylamide gel electrophoresis, or they call it PAGE. And this separates DNA by the fragment size very, very carefully, such that we can distinguish bands or DNA fragments that differ just by one base pair. This sort of polyacrylamide gel electrophoresis is kind of challenging, and this is the original way of DNA sequencing. Now machines have become more sophisticated, such that they can still separate DNA on a 1 base pair difference, but instead of running 4 different reactions in 4 different lanes, as we see here, they run them in just one lane. But what they do is they add DT NTP, so that those are the DD ATP, so like the adenine missing the hydrogen or the oxygen, and the cytosine and the guanine, all in one reaction. But that those base pairs also have attached to them some sort of a fluorescent compound that doesn't inhibit with the PCR. The PCR can still happen normally, but when you hit them with a laser, the laser will recognize a color instead of a base pair. So in this example, a red color coming out is thymine, because the thymine DD-NTP is red. So in this reaction, we're giving a primer, just like PCR. We're giving a DNA template. That's what I'm circling here, this template of unknown sequence. We want to know what the sequence is. So that's going to be something made out of DNA or even RNA. We're going to run a sort of a PCR with it. We're going to include all 4 DNTPs. So these are the deoxyribonucleic acids. They're the building blocks for DNA. We're also going to include all 4 deoxyribonucleic acids. So these are the modified nucleic acids that will truncate the DNA. They will stop it. But those ones also have a different color attached to them. T has red attached, C has blue, A has green, and then G has yellow. And again, in order to actually analyze this data, we need a special machine, and they call that a sequencing machine. So you need access to something like that. All sequencing machines, even to the modern day, even with the Illumina, you name it, require this sort of technology. DNA is fed through a capillary, so that's a gel matrix that is designed to separate them based on size, like everything we've seen, but just one base pair at a time. A laser is passed through that, and it can recognize the color of the band passing past. So, for example, here, based on the color, we can infer the sequence. So blue is C, yellow is G, and so on and so forth. Here's a real sequencing result that I created in my research. So you can see the sequence here, and then that's a sequence that I found in the database that you can find online. And once sequencing the samples, you can find that actually I found that the color or the base pair that corresponded to that position actually was different based on what they call the reference sequence is. So if you're sequencing your own samples and comparing them to what's in the database, you might find there's actually differences. And if it's just a one base pair difference, they call it a SNP, or a single nucleotide polymorphism. So each time you see a highlighted letter, it is actually a SNP. So I wouldn't be, in your own research, don't be surprised if you sequence your sample and find a number of SNPs or differences. Doesn't have to be one base pair. It could be like a string of base pairs that are different from what's in the database. Because plants and animals are very different, even in the same species, have a number of polymorphisms or differences, and that's the point of running these sequencing reactions. You're trying to find these differences, and then some of those differences will actually matter in terms of your trait of interest. And you can use those for breeding or for selection or for gene identification. It's really versatile. So I think that hopefully introduces our next topic of molecular markers. Molecular markers are capitalizing on this concept of linked inheritance. And that means that in DNA, there are genes, there are sequences that really do change the trait. They make the high yield, they make disease resistance, they change the size of the fruit, the taste, you name it. DNA is ultimately where these differences are occurring. Sometimes the differences are occurring by environment, so we use other breeding methods, non-molecular methods, to determine that. But assuming that our traits are determined by DNA, then that can move into the realm of molecular genetics, which is what we're discussing right now. Not all traits are molecular. Some are environment, so environmentally influenced, which is outside this realm. So molecular markers are like really useful because if we use statistics to correlate the marker's presence with the presence of a trait or lack of that trait or marker, we can identify a QTL, or quantitative trait loci. So these are positions within the genome that correlate to our trait of interest. And if we use these markers in a real breeding population, we can speed up the process to incorporate these traits into whatever breeding populations you want. There are so many molecular marker systems. It's a whole topic on its own. I've just had a list of them here. So there's RFLPs, AFLPs. So these are amplified fragment length polymorphisms, simple sequence repeats, SSRs, cleavage amplified polymorphic sites, rapidly amplified DNA polymorphisms, and etc. Single nucleotide polymorphisms. And again, these are all the marker systems, hopefully not to confuse you, all do the same thing. They all do the same thing, in which case is this star here is what we're looking at here is a chromosome. This can be a plant, this can be an animal, this can be a pathogen, a fungus, a bacteria. The star is the real gene that controls that trait. These black lines are like markers, or like that can be any of these systems that you have detected. So they could be the bands on that gel that we were looking at earlier. But the bands, maybe there's some differences, maybe there's something very similar. But here, this blue one, if we run some statistical analysis, means that there is some association between the presence of that marker, so on that DNA gel, that PCR product, if we compare that to whether or not the plant is resistant or susceptible or high yield or low yield or drought tolerant or not drought tolerant, you name it, the trait, if there is some connection, then that's why this is colored blue. And in general, the marker is correlated to the trait because it is in close proximity on the chromosome itself to the actual gene that controls it. Now keep in mind, it might not be the real gene. It's just a marker. It indicates that that trait is probably present, but it's not 100% accurate because it's just close. And it's because those two loci are linked. It's because in subsequent generations through breeding, usually things that are close together on a chromosome get inherited together. And but that biological phenomena is useful for selection. So just as an example of a molecular marker, I'm just including this example of SSRs, or called simple sequence repeats, which are very common in most organisms. So for example, here in one individual, there is a sequence of DNA. This is just an example that is GATA. So it's 4 base pairs, but here it's repeated actually 6 times. So it's GATA, 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 6 times over and over again. And this is a phenomenon that happens when DNA is getting replicated. The enzyme that replicates DNA, if it hits a repetitive region, will just sometimes duplicate it. So this is a very frequent polymorphism. In this individual here, that GATA is instead duplicated 10 times instead of 6 times. So if we were to PCR or amplify that region of DNA and then run that in the gel electrophoresis, we would see some different bands forming. In the plant or individual that just had allele 1, we would see 1 band, and that would correspond to just 6. That's 6 repeated sequence. If we were to genotype or PCR the allele 2, so like our different parent or something, we would see 1 band with 10. But let's say we made a hybrid between these 2 plants, and we looked at their offspring. We isolate the DNA out of the offspring, and we run PCR. We would see a collection of 2 bands, because on one chromosome they have allele 1, which has 6 repeats of GATA, and on the other chromosome they have 10 repeats of GATA. If you were looking at a polyploid species, so like blueberries or potatoes or something like that, you might see even more complicated things. But this is a simple example. And again, you can run these instead of running them on a gel where you see bands, you can run them on like sequencing machines where you would see like what they call chromatograms based on fluorescence of the primers. And so here you can see there's a band here at 180 base pairs, maybe another one at 187. So these are all what we would see as bands on a gel. But if these were 2 different individuals, these bands at 340 base pairs are not present in this individual. So that would be like a different band on a gel that we could differentiate. AFLP markers are like a bit more complicated. They're called amplified fragment length polymorphisms. It's another common system. And these rely on what they call restriction enzyme digestion points in the genome. And what I mean by that is there are some enzymes in bacterium that recognize certain base pair combinations, and when they see that base pair combination, they digest that point. So there's an enzyme called MSE1 that recognizes the sequence TTAA. But it has to be in DNA. So it's TTAA on the 5' strand, but it's TTAA on the opposing strand. And when it sees this, it cuts that DNA in half. But it doesn't cut it in half symmetrically. It leaves what they call an overhang. So on one strand, it leaves just T, and on the other strand, it leaves AAT. T. And this means that that free-hanging T, that thymine, can base pair with any free-hanging adenine, or A. So you can supply your own sequence of primer or something that can facilitate that binding. And for example, here where I'm circling is where you've added extra sequence that is complementary to that overhang. So we call that an adapter sequence. And these adapter sequences are used very frequently, even in modern sequencing applications. Again, on this other side, an enzyme called EcoR1, which recognizes a completely different sequence of DNA that is GAATTC, so 6 base pairs actually, not just 4. It can cut, and it yields again another product that has unsymmetrical fragment, where G is just on one side, and then we have CTTAAG on the other side. And again, with this information, we can add our own sequence to attach, and then by adding DNA ligase, a different enzyme, we can covalently link the backbones together. Why is this important? Why am I explaining this? It's because then after doing that step where you've added your extra sequence, you can just make a primer that is identical to your adapter. So the sequence that you added, you know the sequence. And you know the sequence that the enzyme cuts. And if you just add a single extra base pair onto a primer that matches it perfectly, you can begin to do PCR reactions. And maybe you do an extra PCR with extra base pairs. So the same base pairs of the same sequence that you know because of the cuts. And you can generate random PCR products across the genome. And in one PCR reaction, you can generate many polymorphic bands. So you can be genotyping multiple markers just with one reaction. So it's a sort of a time-saving, a cost-saving process. But the interpretation is very complicated, so that's the downside. So here is a AFLP gel that I created, and you can see that there's lots of bands appearing, and this is all in chili pepper, and I'm looking for disease resistance genes, and any position where there's like a black underline are positions where there's some polymorphism. Again, it's difficult to analyze, but you get a lot of information for a lower cost. Again, all the marker systems like RFLP, AFLP, SSR, CAPS, all these have pros and cons, which is probably too difficult to go into detail. Another system that has come online or come available is called genotyping by sequencing, or GBS. There's a few links I've included here. There are very affordable GBS platforms. And that's because actual sequencing costs are going downwards. So this graph here is the price it takes to sequence the human genome from the year 2001, where it cost like millions of dollars in the US dollars, down to like 2016, where it's almost even $1,000, whereas nowadays it's even less. So if you're sequencing a plant or an animal or a fungus or bacteria, the costs are actually, and again, depends on where you are, are that the costs are going down day by day. So GBS markers, just sequence a single individual, doesn't cost that much anymore. And again, cost is usually your biggest limitation. See where I'm at. So it's something worth looking into before you go into like using gels and things, because it might actually be more cost effective for sequencing. You can use sequencing for to look at gene expression analysis as well. So a big thing is to not know about polymorphisms in a gene, but how much that gene transcript is expressed or not. We can typically do this by two different approaches. That's by quantitative PCR, or they call qPCR, or RNA sequencing. And again, that relies on sequencing. But both rely on PCR. qPCR relies on the fact that you extract RNA, you convert the mRNA portion of the RNA to complementary DNA, or cDNA, with the enzyme reverse transcriptase. And if you're working in a eukaryotic organism, so any plant or animal, you use a primer that has poly-T, so it's just T, that binds the poly-A tail of the mRNA, and then you can basically make a DNA, a complementary DNA version of every mRNA. Then you might enrich that cDNA pool, so you might just make a few more copies of each one that you created with PCR, either with that poly-T tail, or you might include primers of interest if you were just interested in one or two, like genes or something. Then you would run those in a special qPCR machine, where you'd run that PCR reaction with a fluorescence marker. So that could be a dye that intercalates into the DNA, or a fluorescent primer. And then the downstream analysis would mean that a stronger signal in earlier PCR cycles, or like earlier on, would mean that there was more starting RNA in that sample, and that most likely means that there was higher gene expression in your organism. So here we can see sort of a typical data set, well, where each color represents a different individual, and then on the y-axis is the number of transcripts, or the intensity of the fluorescence signal, or the signal coming from that PCR product from the dye you used, either that intercalates or something. And then on the x-axis is the number of PCR cycles. And what you get is these curves. So here, this sample has become creates a more intense fluorescence signal very early on in the PCR cycles, whereas these samples here at the end are producing curves much later on. So that means that this sample had much higher RNA that corresponded to your gene of interest. So it must have higher gene expression than these other ones. And again, by comparing these curves to controls, you can get a much more accurate interpretation of gene expression. You can use this same technology to infer molecular markers as well, by amplifying the same sort of DNA. They call it high resolution melting. So you'd use the same machine. The same machine for qPCR can be used for the same analysis. And that is that you do PCR on a DNA fragment. But let's say the DNA fragment differs by just even a single base pair, like a SNP. In this example, G and A are the only differences between these two sequences. And the difference has to be in terms of the hydrogen bonds between the two sequences. So if the polymorphism was G and C, we would not be able to detect it because G and C both have 3 hydrogen bonds. But G has 3 hydrogen bonds and A has only 2. So between allele 1 in this example and allele 2, they only differ by a single hydrogen bond, which is sensitive to difference in temperature. So if we increase the temperature, or actually from about 70 degrees to 95 degrees, we see what's called a melt curve. So that means that if we intercalate or we add a dye to our PCR product and then slowly increase the temperature while constantly scanning that dye signal, we'll see that here at these temperatures, the DNA double helix is still a double helix. It's connected. But at this point here, when we increase the temperature, it starts to melt and the strands separate, releasing that dye. So we see a decay in the fluorescence. And at this temperature here, the decay is basically 0 because there's... But here, this sample has 1 extra hydrogen bond. So the DNA double helix doesn't separate as quickly at that temperature. So by looking at these, what they call melt curves, we can identify polymorphisms. And this is a really cheap way of looking at SNPs. But again, it involves knowing what SNPs to look at, not identifying new ones. So the last topic I want to touch on is gene editing, which has become really popular in the literature lately. And gene editing has a lot of promise, and it's been useful in many organisms. So I think it's really worthwhile to talk about. And that is used to change the DNA sequence by deleting something or adding something. And I think deleting is more efficient than adding. But the nice thing about it is that the changes are guided and result in very specific alterations to DNA that you really want to target. It's not random like mutagenesis, for example. So the most common way of doing this is with the CRISPR-Cas9 system. So CRISPR is a RNA sequence, and Cas9 is a protein. And here in this picture, you can see both of them. So the CRISPR is the RNA, which you can see was represented by all these letters. And the different colors represent parts of the RNA that are important. So these blue parts are like absolutely critical. The red part are parts that you can change to specify to your gene of interest or where you want to edit. And then the black parts are like sort of variable, I guess. And then the sort of outline shape behind this is the Cas9. That's a protein. And that protein is the thing that binds to your RNA and DNA and cuts. So it's kind of complicated. So I think I like to describe this as sort of a GPS connected to a pair of scissors. So the RNA, the guide RNA, is your GPS. It tells you exactly where to go. And then the Cas9 protein is the scissors. And they are connected together. So for example, you have a guide RNA, Cas9. Step 1 is you have to introduce that to your organism. So usually that's by genetic engineering, if it's a plant. So that involves creating a construct like a DNA plasmid thing and actually introducing that into your plant. So that's time consuming. But that includes two parts, your RNA and then your Cas9. And that will target your DNA of interest. The RNA finds the target, the Cas9 cuts the target. And then different DNA repair enzymes that are already present in the cell will then introduce the edit. And again, the RNA is your GPS, and then the scissors is your Cas9. So you can use this for mutation breeding to introduce like novel variation. You can do this by genetic engineering to introduce new variation. Anyway, so it's very complicated. Like again, that's a whole nother area of research. And the last thing I want to touch on is, in Africa, getting hold of these technologies is complicated, and like it sort of assumes that you already have a lab to work with. So I just want to like touch on the fact that resources are probably the biggest limitation. But there are different options for obtaining less expensive lab equipment. And modern innovations are like making equipment less expensive. For example, 3D printed lab equipment is becoming more mainstream. There's examples available of like 3D printed microscopes, centrifuges, magnetic stir plates, mixers, spectrometers, plant tissue growth, so doing like plant genetic engineering, and then even like test tube holders and things. I don't know about PCR machines, but maybe. There's here on the side are like different resources you can probably connect with to see, because they are actually creating these tools for Africa. So openlabware.net, Trend in Africa, etc. And I thought this was like impossible, but I was presented with a challenge once in my career. Like I needed to pour a lot of Petri plates, and so I actually created a robot that could create Petri plates. And so this video here is a robot that I built not by myself, but with the help of people that could pour Petri plates. And we built this for like far cheaper than you could buy. So if a piece of equipment is the true limitation, it is possible to do it yourself. And I know it's not, it is like daunting and intimidating, but it is possible. And there are tools out there to help. So again, here are even more links for open source 3D printed lab equipment, and they're updating these on a regular basis. You can download the templates for free, and again, I don't know all the details for how to do it, but at least it's a way of getting established. And all the methods I've mentioned today, maybe apart from some of the sequencing analysis, can be accomplished on some of these equipments. So like microscopes and centrifuges, and again, if 3D printing or trying to build it yourself is completely out of your skill set, that there are ways of getting very simple equipment. So I would recommend this Bento Lab. It's not free, but it's an all-in-one lab in case you can't do anything like, and that includes thermocycler for like PCR, or restriction digests for like gene cloning, centrifuge, UV transilluminator, and gel box for like gel electrophoresis, and then like a stand and like a pipette. So you can, it's like an all-in-one lab that's, it's a bit expensive. It's like about 1,000 British pounds, which is expensive. But you can look at more about it here. And again, that's again an all-in-one lab in case you cannot like accomplish anything else. So hopefully these are like tools that make your research sound like more feasible if you want to accomplish like molecular like goals. Anyhow, this is just an introduction. There's so much more going on in the molecular world. There's like RNA, so it's like silencing your genes. There's cloning, detecting RNA and proteins. There's genetic engineering, using the markers actually for breeding purposes. I didn't even get into any of that, so marker-assisted selection. So this is just an introduction. Anyhow, thank you so much. I really feel privileged to be helping the GR Biotech Foundation, and I have to thank Carol for organizing all this stuff, and I really hope everyone appreciated this talk. Please get in touch with me if you have any questions.